It's such a pleasure to be here, and I am so honored to be associated with the Spastic Paraplegia Foundation. I've had some funding, as Frank mentioned, from the foundation that's been absolutely essential for me to be able to do this work. And I know that that money does not come easily, and that the patient population is extremely active in earning this money through various events that you do. And I want you to know that each and every dollar, each and every penny of that money that we use very carefully toward hopefully achieving some better understanding, leading to therapies that will help people now and in the future who suffer from these diseases. What I'd like to do is share my screen. What I study is hereditary spastic paraplegia, and this disease is most characterized by gait defects. And there's a rather characteristic walk gait that patients who suffer from hereditary spastic paraplegia display. And you can see an example of it here. The gait defects sometimes are relatively mild and sometimes get to be so severe that the patients wind up in a wheelchair. There's a spasticity associated with this, lower limb weakness. And then sometimes there are other symptoms as well. There's some bladder difficulties and different sorts of other problems associated with the disease. Usually, but not always, usually it's adult onset. The average age may be, depending on the particular mutation, particular gene mutated, average age is probably in the 40s, but we see a lot of people entering their early 20s who begin to display a bit of spasticity or gait problems. Other people really don't display much until maybe their 60s or 70s. It's really quite variable among the patients. Now, what goes on in hereditary spastic paraplegia anatomically is degeneration of something called the corticospinal tracts. The neurons, the cell bodies of the neurons, are in the brain, and they descend down into the spinal cord where they synapse with other neurons. And what happens in hereditary spastic paraplegia is that the axons that project from these neuronal cell bodies that comprise the nerves will begin to display swellings, other indications of degeneration, and then undergo something we call dying back degeneration, where they actually pull away from their synapses and begin to wither away. And this is really anatomically what accounts for the problems with gait, with walking, with other sorts of issues that we discussed. It's not exclusively the corticospinal tracts. There can be some degeneration in other parts, other tracts as well, but this is generally where the problem lies. Clinicians will generally define diseases in terms of their symptoms, whereas research scientists, such as myself, we like to think about the etiology, in other words, the cause, the mechanism underlying the disease. So by classifying diseases by their symptoms, hereditary spastic paraplegias are those diseases that share the sorts of symptoms that I just described. So a physician sees those symptoms and will define that as hereditary spastic paraplegia. But whether spastic paraplegia is really all the same disease, or whether this is various different diseases that just look the same because they manifest in similar symptoms, we really don't know. It's probably more likely that they are at least converging on a common pathway, but not necessarily. So this is why there are different researchers who are looking at different genes that produce this particular disease. Now, here's a graph that you can see some of the different types of hereditary spastic paraplegia, SPG3, SPG11, and these are defined in terms of the particular gene whose mutation is causing the disease. As you can see here, the most common by far is SPG4, and that's the one that I study. So this is the most common one. There are also, though, a whole lot of patients who have hereditary spastic paraplegia with no gene mutated thus far identified. So the list of genes whose mutations are associated with this disease now is about 83. When I first got into this several years ago, it was about 12. And now it's about 83. So there's a growing number of different genes whose mutations cause this disease, or at least are associated with this disease. Over 40% of the cases, at least the cases where the mutation is known, is SPG4. And the SPG4 gene now is called Spast. Either one is fine. Spast is the more modern name. Now, when I say mutation, what is it that I actually mean? A mutation is a change in the genetic code. So you see here chromosomes, and as you know, chromosomes are made up of DNA, and DNA has a code, a sequence. It's 4 different bases that alternate and comprise the genetic code for an entire organism. When certain of these bases change, and they can change sporadically, or they can change in response to, say, some mutation or some radiation, I should say. They can change for all sorts of different reasons, some of which is just random and sporadic. The vast majority of mutations that'll happen are really harmless. They don't really matter. Some mutations are so bad that the organism just can't live and dies. There are some mutations, though, that are not lethal, but they're also not harmless, and they cause diseases. So these diseases, once they become part of the genome, they will then be heritable. So let's say even if a person just sporadically acquires this mutation, then that person now can pass that on to his or her children. Now, some mutations that cause diseases are sex-linked. That would mean they're associated with the X or Y chromosome that decides the sex of the human being. Others are what's called autosomal, meaning it's just not part of the sex chromosomes, but just part of the other chromosomes, the ordinary other chromosomes. And the SPG4 is autosomal. Now, it could also be dominant or negative. Now, this means that if one of your genes has some mutations, if it's a dominant situation, you only need one of them to be mutated, and then you've got the disease. If it's recessive, that means you've got to have both of them be mutated. Otherwise, you're fine. As long as you have one that's good, you're fine. So what SPG4 HSP is autosomal dominant, meaning if the individual has one of the genes mutated on one of the chromosomes, you've essentially got the disease. That's a little tricky, though, because we really don't know how many people might have this mutation and display no symptoms at all. That's because of that variability that I mentioned in the age. Let's say the individual would begin to show the symptoms once he or she becomes 80 years old. Well, maybe that person passes away at 75 and never shows the symptoms because he or she is just simply not old enough to display them yet. Some people are just not complainers. If they have mild symptoms, they may say, oh, I don't have the disease, I'm perfectly fine. But if a physician studying HSP looks at the individual and sees signs of that characteristic gait deficiency, then maybe that person does have the disease. So the diagnosis gets a little tricky. Now, what I want to do is take just a few minutes and talk a little bit about the Spas gene, because this is really how I got into this. What the Spas gene encodes for, each gene encodes for a protein, and the protein is called spastin. And as Frank mentioned, I study microtubules. This is what I've studied for 35 years, this cytoskeletal element in cells called a microtubule. And what spastin is a microtubule severing protein. It sits on the surface of the microtubule. You see it here as this hexamer. It sits on the surface of the microtubule and it breaks the microtubule. Now, before you think that's some horrible, awful thing, no, that's physiological. This is important because long microtubules need to get broken up into short microtubules so they can be deployed into the axon or the dendrite so that they can assist in the formation of branches from axons and dendrites. So this is a normal ongoing process of microtubule severing, and this is the work that spastin does. Now, what microtubules are cytoskeletal elements. They populate axons and dendrites. When I say axons, I just mean the fibers that make up nerves. And what they do is they provide a couple of functions. One is architecture. They're the 2x4s, the elements that give structure. Without the microtubules, the axon would just collapse on itself. So it's the structural elements. But the other thing that it does is it provides railways for transport. So all sorts of different organelles like mitochondria and other sorts of organelles need to move up and down the length of the axon. And microtubules are the railways along which that happens. So here schematically, you can see what are called motor proteins that are latching on to cargoes and then walking along the surface of the microtubule in order to convey what's called axonal transport in both directions. So microtubules are really important is the point, and they have to be regulated properly. Now, a little bit about me and how I got into this, because my training is not as a medical doctor, and my training is really not as a pathologist. My background is not in disease. So how did I get into this? I did my graduate work at Michigan State University and then my postdoc work at Temple University. And right from the beginning, I was always fascinated with microtubules and neurons. So this is what I've always studied. How do the microtubules get organized in neurons? How do they function? How are they regulated in the advance of the axon, the growth of the axon? How do dendrites become different from axons in terms of their microtubules? So from my graduate work to my postdoc work, and then for my faculty work, for the first 10 years, I was at University of Wisconsin-Madison. And then ever since, for the past 20 years, I've been at Drexel University in Philadelphia. And throughout this work that I've done on microtubules, I've been funded primarily by the National Institutes of Health for this work. And there's a promise that's made by basic investigators when we request funding from NIH, that if NIH is to fund us on all of these things that we find interesting about how axons develop and microtubules get organized, that one day this would really be important for human health, disease, injury issues. NIH made for decades an enormous investment in basic science, and really it was a very wise decision. As you know, government organizations are always wise in their decisions, but this was a very wise decision because now we have an abundance of knowledge that really is helpful in resolving these questions related to human disease. So I was studying microtubules and microtubule severing and how important it is. And then around 2002, it was discovered that the spastin protein is a microtubule severing protein. Before then, spastin was simply known as the gene product of the most common form of hereditary spastic paraplegia. Nobody knew what it did. Nobody knew what its function was. So getting under the hood and figuring out what we could possibly do when this protein malfunctions, when it's mutated, was really not possible until that discovery was made. And then when that discovery was made, suddenly I became one of the most qualified people to investigate the disease, and I've been involved in it ever since. So again, microtubules, they are polar filaments. They have a plus end and a minus end. They assemble, they disassemble. They act as railways for transport, as we just talked about. And both long and short microtubules are really important. Some have to be long to provide these railways that we talked about, and others have to be short because the short ones that are most mobile, that can move around, can be reconfigured for the plasticity of the nervous system, which is important, obviously during development, but also in the adult, that the nervous system has to be plastic. So where do the short microtubules come from? They come from these proteins that chop them, like spastin. There are locations that this is particularly active, growth cones at the tip of the axon. Sites of branch formation is really a site where this becomes very important. Now, here you can see experimentally what we've done is we've taken a very simple cell. This is a fibroblast that you see on the right. That's just an ordinary fibroblast, and you see fluorescently the microtubules. So we have a fluorescent antibody. This is a way of making the microtubules glow so you can see them. Now on the right, you have a cell in which we've used a genetic approach to overexpress catonin. Now, catonin is very much like spastin. We first started studying catonin. So you can see just how avidly that it breaks up the microtubules. Here's spastin, and in panel A, you see again the control, and then panel C, you see the overexpression of spastin, and you can see it's really effective at breaking microtubules. So through this work that we did a few years ago, or now maybe 10, 12 years ago, we really began to understand this protein and how it works in the nervous system, the healthy nervous system, the developing nervous system. We found that this is really important for branch formation, so that if we overexpress spastin, we get more branches. So these are cultured neurons. These are actually rat hippocampal neurons. And you notice in the top panel, you have a few branches, but if you overexpress spastin, you get a lot more branches. So this really fortifies our thinking that spastin's important developmentally for branch formation. Now that we understand a bit about this protein, what can we do in terms of understanding the disease where this protein goes awry? Now, what I want to do is explain something that's really important about disease, and that's loss of function versus gain of function. When a protein is mutated, oftentimes it no longer works properly. And that's how we understand the disease, loss of function. Well, say take with spastin, for example, if it's mutated, now it can't sever microtubules. So you've got one good spastin gene, you've got one bad spastin gene, and now you've only got half the ability to sever microtubules, half the ability to do so. That's sometimes called haploinsufficiency, when half as much isn't enough. Sometimes half enough is enough, but if it's not, then it's haploinsufficient. And that's just simply not enough of the appropriate function. But what's interesting is that in a lot of neurodegenerative diseases, it's more common for the disease to be caused by gain of function. Now, gain of function might sound kind of like a good thing, but it's not. Gain of function means that mutated protein gains some new function, some new ability to do something it's not supposed to do. And that's not good because the protein is now doing strange things. And the way that this can be understood very easily is loss of function is the kid didn't show up for class. He's just not there. And, that's not good, but it's not horrific either. But gain of function, he's there, but he's making trouble. And this can go on and get pretty bad if it keeps going and accumulate more of these problem students, and they're making more and more trouble. That's gain of function. So their students are there, but they're causing a lot of trouble. In the HSP field, when I first got into this, which is around 2008 or so, nobody else in the field pretty much knew, they weren't from the microtubule field. And for genetic reasons, most people thought that the disease was caused by haploinsufficiency. But when I got into this, it seemed to me that there were some problems with the haploinsufficiency model. Why was the disease only in the cortical spinal tracts? If spastin's really important for severing microtubules throughout the body, throughout the nervous system, why does the cortical spinal tracts really show this specificity for degeneration? Why no developmental abnormalities? That's where spastin's really the most important, as I just showed. But generally speaking, as I said early on, the age of onset can be 40 years old or 70 years old. And it seems like half as much spastin function is perfectly fine. It's perfectly fine for the development of the nervous system, for the fetus, for the child, for the teenager. So why all of a sudden is half enough spastin not sufficient later in life? And there are also some mutations that actually don't lose their function because the mutation is in some other part of the protein, and they seem to sever microtubules just fine. So could it be a gain of function mechanism? And this is the hypothesis that has driven a great deal of what I've done. Now, in the world of science, don't let this cartoon bother you too much, but in the world of science, there's a lot of debate that goes on. And when I entered the field, the idea of gain of function was not seen as the more likely explanation. Let me put it that way, that most people in the field were quite invested in the haploinsufficiency model. And they had their reasons. I mean, their reasons are valid reasons. But I introduced this other way of thinking that maybe this is gain of function. And this took a little while to really solidify in the field. But let me tell you some of the reasons and some of the thinking here. This slide is very complicated, but what I want to illustrate here is on the top, we see the full-length spastin gene. And on the bottom, we see what's a shortened version, just a little bit shortened version, because it turns out that this gene has what's called two start codons. So the start codon is where the gene becomes active in making the protein. And for some reason, what generally happens for almost all cells of the body, for almost all times in your life, the spastin just skips that first start codon and makes this shorter, somewhat, not a lot shorter, but a little bit shorter protein called M87. The longer protein called M1, you just really don't see much anywhere except one place. And I'll get to that in just a moment. But the idea of this M1 protein is that the M1 specific region is very hydrophobic. And because it's very hydrophobic, it tends to misfold. And what misfolding is a protein, by virtue of misfolding, will become very stable, will accumulate, and parts of that protein that normally are kind of buried in the molecule become exposed. And those are your troublemakers that normally are not exposed, they become exposed. And now that misfolded protein won't go away, it accumulates, and it makes trouble. So this is the idea that maybe it's M1, maybe it's M1. Now, I said that you only begin to see M1 in one place. This is called a Western blot. And the Western blot is just a biochemical way of looking at proteins. And this band that you see at the bottom, this is the M87 band. It's everywhere. And the M1 band, which is up here, you don't ever see much at all except for in the adult spinal cord. This is spinal cord. And remember, the spinal cord is where the corticospinal tracts are located. So for some reason, those axons tend to accumulate M1, even wild type, even regular M1. They tend to accumulate a little bit of it. Maybe they need it for something. Maybe they need extra. But on the other hand, maybe they're a little bit, those axons are a little bit more challenged in terms of what's called autophagy, of processing and getting rid of proteins. So it tends to be a little leakier in terms of that accumulating. And that's really where our hypothesis lies, that because the corticospinal tracts are where M1 can accumulate, that they become a trouble place for the mutant M1 to accumulate. So over the years, we worked on various different experimental models. And among the first models that we worked on is rat neurons in culture, where we culture these cells, like I showed you early on, where we cultured hippocampal neurons or cortical neurons, different neuron types. And what we would do is make these mutant genes using molecular biological techniques and then express them in these cells. And these are just some of the students who've been in my lab over the years who've done this work. We've also worked with colleagues, this is Dan Miranda, and we worked on Drosophila. And whether it's the cells, rat cells, or Drosophila, if we express the human mutant M87, it's really not much of a problem. So here's the wild type fly, the ordinary fly. And if we express the mutant M87, notice the wing is a little bit goofy, not bad. The fly flies fine and it flips around fine. But here, the fly is expressing the mutant M1. And notice that it's holding its wings very strangely, and it can't fly. If you turn it on its back, it can't flip over. So this, and also the cells from the rat, it's the same result through and through, that the mutant M87 is not that bad, but it's the mutant M1 that is. Now, we also worked with squid. Now, you may wonder why we're working with squid. This is the work of our collaborator, Gerardo Morfini. And what's great about the squid is they have a giant axon that you can squeeze out like toothpaste, and you can really do a lot of studies on axonal transport. And if we infuse mutant M87, there's no ill effects on axonal transport, but if we infuse mutant M1, there's significant deficits in axonal transport. So all of these observations really fortify the view that the mutant M1 is creating some kind of gain of function toxicity that's detrimental to the health of neurons. And that, we hypothesize, is really at the heart of this disease. And now that we know that, we can begin to look at mechanism and therapy. For example, with this axonal transport work, we know that all of these axonal transport deficits are rectified by inhibitors of casein kinase 2. And that kinase, like other kinases, is responsible for phosphorylating proteins. And protein phosphorylation is just a way of turning on and turning off function. And if what the mutant spastin is doing is aberrantly activating a kinase, you can certainly imagine how that would cause a lot of problems, because now the phosphorylation status of various proteins is abnormal. Moreover, we can begin to think about therapy. We can begin to think about, if that's a significant cause of the problem, can we use these inhibitors of CK2 as medicine to help alleviate the symptoms in patients? What we really lacked at that point, though, was a vertebrate model. A vertebrate model, usually mouse is what people use. A vertebrate model is really important because in order to get to clinical trials, you have to do preclinical work on an animal. And a fly or a neuron and cell culture or squid exoplasm is not going to show a gait deficiency, for example. So how do you demonstrate that your therapy is actually rectifying the gait deficiency if you're working with extruded squid exoplasm? That's not going to work. So we decided to make a genetic mouse to model the disease. Other investigators at this point were using what's called a knockout mouse that has no spastin. They knock out the gene, knock out both of the spastin genes. Why would investigators use a knockout mouse to model the disease? It's because their model, their mechanistic thinking is haploinsufficiency. If they're thinking the reason for the disease is that the spastin protein doesn't work, then it would be perfectly modeled simply by making an animal that doesn't have any spastin. What's interesting, though, is that the knockout animal really doesn't show all that much of a phenotype. By phenotype, I mean, does it have a gait deficiency? Does it have spasticity? And the answer is no. It's quite amazing that you have this animal that has no spastin, and it really doesn't show much by the way of defects. There's a few things you can document, but really not much at all. It's probably because when there's no spastin, then catonin or some other protein can pick up its duties. So what we wanted to do is to specifically model our thinking, which is the gain of function hypothesis. So the approach that we used was to take the mouse and allow it to have its own spastin genes. We're not going to take away those genes, but we're going to add to it. We're going to add into a different chromosome location human mutant spastin that causes HSP in humans. So these animals that we're creating in this fashion are not haploinsufficient. They've got all their endogenous, their normal mouse spastin. They can sever microtubules just fine. But we're adding on top of that the human mutant spastin. So these animals are not haploinsufficient. They do have the potential for the gain of function effect. And this work is done by Liang Chong and Emanuela Piermarini. It's two very excellent senior scientists working with me. Now, I want to show you what we came up with. This is a wild type animal. When we set forth to make our mouse, we originally didn't know if we would have to target the expression of this human mutant protein specifically to the motor neurons that go awry in the disease. We figured, well, if we express it everywhere in the mouse, is it just going to kill the mouse because we think it's a toxic protein? But what's very interesting is we started out that way. We started out just by expressing throughout the mouse, and the best possible result would be that just like the human disease, somehow here the disease afflicts the corticospinal tracts. So let me show you this mouse. Now, I want you to keep in mind that the knockout mouse would look identical to this mouse. I could easily just trick you and show the same movie again and say, this is the knockout mouse, because the knockout mouse looks just like this. But here's our mouse. This mouse is alive. doesn't show a lot of other problems, but look at its gait. It can really barely walk on this beam. So this is quite remarkable, I think, that this animal is not haploinsufficient. The haploinsufficient or the complete knockout animal walks just fine, but an animal that has the gain of function component displays a remarkably similar gait deficiency to the human patients. And it's adult onset. You don't see this until about 3 months, which qualifies as adult for a mouse. Adult onset, gait deficiency. It also kind of shakes in your hand a little bit like it's spastic. So it really shows these characteristics. And then here's just a lot of quantification of these behaviors. But how about this dieback phenomena? Remember I said that there's a couple things that happen anatomically with the disease. One is the axons get these swellings, and the other is that the axons die back. Now, in order to see if they die back, what we do is call these tracer studies, in which we inject a dye into a certain level of the spinal cord, and then we look and we see if the axons don't go down as far, to see if they're pulling back. And what we see with these couple of qualities, both the swellings and the dieback, is the swellings. This is actually very interesting. The swellings, the knockout mouse does show them, but ours doesn't. Our mouse shows the dying back, whereas the knockout does not. So our mouse shows the dieback and the gait deficiencies, whereas the knockout shows the swellings, but not the gait deficiencies. So what we think is going on here, I know the slide's a little bit complicated, but what we think is going on here is that the gain of function component, this toxic M1, that's producing the degeneration that really causes the disease. That's enough to cause the disease. But the haploinsufficiency will not cause the disease, but it's causing these swellings that are making the axon more vulnerable to a second hit, which is the gain of function. So you put them together and you've got a more severe degeneration than you otherwise would have. And that's what we're thinking, that gain of function and loss of function both have a role to play. The gain of function is really causing the disease. The loss of function is not, but the loss of function is exacerbating the disease. And that's very important because remember I said there are some people who maybe they can live until they're 70 and they show no symptoms. If we can treat the loss of function component of the disease, maybe that individual can get 10 more years of high quality of life without treating the gain of function disease. The gain of function component of the disease, I do think is the one we want to focus on. But the loss of function component is a viable component for treatment. Now, there are some people who will say, well, everything you said makes sense, but there's one thing that's really bothering me. And that's that there are some patients who have this disease, SPG4, HSP, who have a complete gene deletion. They don't have a mutant protein. They don't have any protein, and they have the disease. How do I explain that? Because they should be like that knockout mouse. They shouldn't have the disease, if my thinking is correct on this. The answer to the question is I don't know. I don't know what accounts for that, but I have a pretty good idea. Remember I said earlier on that there are over 83 genes whose mutations cause some version of hereditary spastic paraplegia, and that list is growing all the time. My guess is that there are some people out there who have a complete deletion of the spastin gene, and they're perfectly fine. They're perfectly fine. But how would we know? They would never come into the clinic. They would never have their DNA analyzed. The people who have the gene deletion, who have the disease, I'm going to hypothesize have another mutation to another gene that hasn't been identified yet. And that maybe, if that gene is mutated on its own, maybe it does not cause HSP, that the spastin gene has to be mutated too in order for the axons to display those swellings, in order to be vulnerable sufficiently for the second hit to be effective in causing the disease. So it just goes to show how complicated disease can be, and how, when it comes to this disease, by no means am I arguing that we should ignore loss of function, because I think both of them are really quite important. How do we test this? How do we test this idea that the gain of function, loss of function are contributing, and the loss of function is making the animal more vulnerable to the gain of function? Well, we can cross these animals. So we can take the loss of function animal and cross it with the gain of function animal and see if the symptoms are actually worse. So we're using, for the behavioral analyses, we're using a more sophisticated approach than the beam walk that I talked about a moment ago. This is called the catwalk. And what's really interesting about the catwalk, powerful about the catwalk, is that you put the mouse through this corridor, and a movie is made of the animal with the paws landing, and then the data collected can be analyzed in a huge number of different ways to see what differences might arise. And here's, you can kind of see a little schematic here, how it works. So every time one of the paws comes down, then data can be collected. And indeed, what we found, you can kind of, if I can follow this a little bit, that the animal that is the cross, that has both gain of function and loss of function, represented by the red bar, is always worse than either one alone. So this fortifies our hypothesis that both are contributing. And I think this is really important to know, because I do believe that there are benefits to be accrued in treating each one of them. So when it comes to treatment, now we can begin to say, well, what are the mechanisms underlying loss of function, and what are the mechanisms underlying gain of function? So how can we treat them? So with loss of function, for example, if there's not sufficient spastin to sever the microtubules, what problems is that going to cause? Is that going to cause flaws in the organization of microtubules? And if so, how do we correct that? Now, this is something I've been studying for 35 years, as I noticed, so we've got a lot of good ideas about that, I think. The gain of function, a few strategies. One is this CK2 I mentioned, this casein kinase 2 aberrant activation. We can use inhibitors of CK2 and see if that helps. The other thing we can do is try to get at the heart of the accumulation of the mutant M1. So we can try to enhance autophagy. Autophagy is the process by which these misfolded proteins are cleared, and drugs like rapamycin can be used to enhance autophagy. So by having knowledge of these mechanisms, now we can really tap into a whole lot of information that we've gathered over the years to be able to treat each of the components of the disease. And this is a schematic that Dr. Morfini and I came up with together. This photograph you see is Dr. Cheong, and he's an expert on human-induced pluripotent cell technology that you've already heard a little bit about in the previous talk. And what's going on here is that he's taking fibroblasts and making them into induced pluripotent cells, and then introducing various mutations of the SPAST gene. Another thing that's really interesting about this disease is that there are over 200 different mutations of this gene in different patients. And are they all the same? Depending on what type of mutation it is, what domain of the protein is mutated, is it all the same, or are different mutations causing different problems? So one of the nice things about this technology is that Dr. Cheong can make cell lines that are absolutely identical, with the one difference being the mutation is different. These are called isogenic cell lines. So you can compare them all directly. If they were all, say, from different patients, you wouldn't quite know because they have different genetic backgrounds. But by keeping everything the same and just varying the mutant, the particular mutation, then he can do a lot of studies to compare and contrast. So these iPS cells can be differentiated into motor neurons. They can also be differentiated into glial cells. You know, if any other cell of the body might also be contributing to the disease. So they have a great deal of power. Now, these are those swellings that I mentioned. And with these, you see these swellings that reflect the human disease. And in the upper panels here, you can see these swellings. And what we've done here in the lower panels is we've treated with a drug. Now, what this drug does is let's just say that speeds up the movement of the microtubules. So if the microtubule movements are sluggish because there's fewer short ones, well, how about if you get those fewer short ones moving faster? Is that going to resolve these swellings? And it does. At least in cell culture, it does. So this is just an ideal example how all of this basic science knowledge we've gathered over the years can really be utilized in order to rectify these symptoms, and then hopefully the behavioral problems that patients suffer. So really, at the end of the day, when you're looking for the right therapy, you need the right mechanism. If you've got the wrong mechanism, then you're going to spend a whole lot of time on the wrong therapy. And we see this all the time, say, with Alzheimer's disease, that the New York Times publishes that there's some big clinical trial with some sort of new drug, and you read a little bit what that drug is supposed to do. And I think to myself, oh, that won't work. That won't work. And after $1 million in a couple of years, guess what? That didn't work. And I think taking a wiser approach toward therapy is really going to be very hopeful with spastic paraplegia. So the lessons here: know your mechanism. Basic science, I think, really is the key to the answers. Therapy requires the right tool to fix the problem, and you also need good animal models in order to test the therapies. And I think we've developed a good one. So I want to acknowledge all these different people who've contributed to this project over the years. So here's a list of their names. And I also want to acknowledge the Tom Wallach Foundation, which provided me some funds. And in particular, the Spastic Paraplegia Foundation, which, as I mentioned at the beginning of my talk, has been so supportive and provided us some funds, but also as a support structure to keep me in contact with patients and with the community, and to provide a lot of moral support. It's been invaluable. So I particularly want to thank Frank and Norma and everybody associated with the foundation. And I want to thank you for your attention. And I'll stop my screen share. Let me see if there are any questions. Frank mentioned earlier to me that there would be a separate opportunity to answer more questions. So I can look in the question box. Someone's asking, Laura is asking, how does CK2 inhibition rescue axonal transport impairment? Well, the way that CK2 inhibition rescues axonal transport impairment is that the mutant M1 protein by hyperactivating CK2 activity, the CK2 aberrantly phosphorylates molecular motor proteins. So remember I told you very early on, I showed you the slide, and I said that these motor proteins are walking on the microtubules to convey cargo up and down the length. Well, when the mutant M1 aberrantly activates CK2, which aberrantly phosphorylates the motor proteins, that doesn't happen well enough. So the CK2 inhibitors can reverse that process to get those organelles moving again. Now, therapeutically, the problem is that the CK2 inhibitors are okay for squid axoplasm, but they're not quite specific enough to enter into the human being. And also, you wouldn't want to completely shut down your CK2 because it has work to do. So that's going to take a little effort in order to get that finding into something that's viable for therapy, but we're definitely working on it. So Kevin asks, are you doing any clinical trials? Not yet, Kevin. I mean, we've got the mouse, which is that ever important first preclinical step, but clinical trials for the CK2 or the rapamycin or some other ideas that we're pursuing, I'd say clinical trials are at least a year away. But the good news is that our trajectory is the right one. I believe it is. That if the trajectory is the wrong one, you can have all the clinical trials in the world and it's not going to get you anywhere. So I feel optimistic. I feel optimistic about it. Any other quick questions? Okay, I think that was all the questions, but I'm really happy to entertain any further questions if if any of you want to attend the future event that Frank's going to arrange. And then also feel free to keep in touch. I mean, my email is readily available, so feel free to keep in touch. I really like hearing from patients. So thank you very, very much for your kind attention.